Rest.
All right, should be ready to go, huh? Well, thanks again, everyone, for joining this evening. We have a little competition tonight, as you know, probably with Hawkeyes playing the second game in NCAA. I'll try to get through this quick enough so you can watch the end of it, see how they come out at the end. I got a few requests, basically, to get this. This one's basically set up on the requests that I received or Lance received from some of you folks in terms of what you'd like to hear me address. I did throw in a couple of things just myself. So that's what we're going to be taking a look at. It's kind of what the audience wanted to see here. So the biggest question that came up from a couple of people was help with reed canary grass in terms of distinguishing it, mainly, of course, vegetatively. I think most of us can probably recognize reed canary grass. Let me get my pointer here. Reed canary grass, you know, when it's fully developed and mature, especially when it's in a dense stand like this. So, you know, that's usually not too much of a problem. The vegetative, you know, without it in flower or fruit and younger forms, you know, could be a little bit more challenging. It certainly is an invasive species, but we have to remember, of course, that reed canary grass is native to North America. It's actually one of 5 species of Phalaris that are native to North America. We also have 6 other non-native species of Phalaris in North America. And it's probably mostly native to the northern, no one really knows for sure, but probably maybe kind of from us here in Iowa on north. So like the northern part of North America, the northern half of North America or so. You know, it's really very hard to tell for sure because the European version and the cultivar version, which we have and is the aggressive invader, you know, of course, is very similar looking in many ways to the native one. There's been research done in Minnesota recently. I found that they utilize DNA, which of course is the modern way of now of figuring out where plants come from and what a plant is. They used DNA and they took DNA samples from 6 populations in Minnesota from pretty much riparian habitats, meaning along rivers. And then they also looked at 6 riparian populations in the Czech Republic and Europe. And what they found when they looked at the DNA was that these samples came out so that they were clustered separately. All of the Minnesota samples clustered together in terms of similarity in their DNA, and all of the European ones clustered together, but in a different place, basically in the figure that they made, indicating that these were 2 distinct, you know, DNA groups, if you will, which suggested then that at least the samples that they had looked at in Minnesota were not European. And so they were native. So again, no one really knows really just how much and where native strains are. I mean, I think I've seen some in Iowa here and there. If you see really dense, aggressive, vigorous growing reed canary, it's probably the non-native one, because again, that was developed as a cultivar in Europe to be used in erosion control, to be used in waterways, probably maybe a little bit of forage, but mainly to control erosion. And so that very aggressive and vegetative growth habit that we see, that was a part of the plan. But it is, of course, a very, very aggressive and invasive species, especially in sedge meadows or wet prairies or on floodplains, any place where it's somewhat wet. So we do have to be able to distinguish it. So this slide here just kind of shows some of the basic characteristics of reed canary grass. And, you know, what we see down here in the lower left here is a nice photograph of the Really important parts of the grass are right at the point where the sheath, which wraps around the stem, starts at a node and then goes up the stem, up across the internode to some point, and then the leaf blade then comes off the top of the sheath. And this is where the ligule is. There's lots of important structures, of course, right in this area. And so we see this very, very distinctive, somewhat distinctive, very large membranous ligule. That's characteristic of reed canary grass. It's usually in the range of 5 to 10 millimeters or so in height, so very conspicuous. This collar is just this area that's kind of light whitish yellow. There'll be another slide here. Let's see, here's another slide here. This is the collar again area. The collar is basically this area between the blade and the sheath. And so it's again where the sheath and the blade meet. And again, in reed canary grass, this is usually fairly distinct in terms of its color, this lighter yellowish color that we're seeing here. There are no auricles, there are no projections that come off the collar in reed canary grass. The nodes can be pretty much the same uniform color as the stems, or sometimes the nodes are more. Here's a node over here that's fairly light color. Again, it looks like the collar. The leaf blades are fairly broad, fairly wide, and that's going to be one of the most distinctive things to help you separate it from other grasses that could be in the same kinds of environments, in wet meadows and wet prairies. The width of the blades are usually from 5 to 20 millimeters, but that's the range it can encompass. But usually they're well up on the high end of that. So usually they're at least 15 millimeters or more wide. Again, typically full grown and mature plants. The inflorescence, the secondary inflorescence, the inflorescence that has all the spikelets open and spreading here in this form, but this is the form that it's in when it's in anthesis, when it's shedding pollen and it's also accepting pollen. When it's first, when it's first emerging from the boot of the grass, like these right here, you see the secondary inflorescence is much more contracted, very much closed and more contracted. Of course, then it opens up more so when it's ready for pollination. And then it will, again, sort of contract later on. This is an inflorescence here later in the growing season after pollination is done and seeds have formed. And it, again, kind of contracts back down somewhat. So the thing that ultimately will always identify reed canary grass, of course, is if you do have the flowers. We want to focus a little bit more on vegetative aspects, but if you do have the flowers, you have the spikelets. That's what we're looking at here and here. These are the structures that are called glooms. We've talked about these last week, of course, with wild rise. The glooms are these two bract-like structures that sit at the base of the spikelet, which again is the inflorescence, the primary inflorescence of a grass. And then above these two glooms is a floret, a flower. There's actually 3 of them, but it looks like there's really only one, because 2 of these florets, one right here and one right here, are thought to be the result of reduction, and they're sterile, as it's pointing out here, so they're non-functional. But they still sort of exist as a tiny reduced lemma. Again, one of the other structures on the grass that we talked about last week. And so you can look at these and see them as sort of these bristles, I suppose, hairy bristles, but they are, at least developmentally and morphologically, thought to represent very, very reduced lemmas. Which again, remember, there's a lemma for each flower. This is the mature and robust lemma right here for a flower, the lemma here and the palea here that enclose a fully functional bisexual flower. We can't see it because it's in between these two things here, but it's these two sterile florets that really, again, set Phalaris off from all other grasses. And here we can see, again, another, this is just a different picture from a different web source. But again, those two arrows are pointing to those two sterile florets, which are pretty easy to see if you just collect a little bit of an inflorescence here, you know, and take it back and look at it under a scope. But again, we don't really have to do that. We don't want to have to do that. We want to be able to identify these reed canary grass vegetatively if possible. So there are some other things to look for here. As I said, very broad leaves, of course, very robust plants, very distinguished collar, you might say, distinctive collar, very tall ligule. Now, I do not find any reference to this in all of the sources that I looked for, but I have looked at hundreds and hundreds of reed canary grass plants that are emerging from seed in seed bank studies. I did a seed bank study down in Madison County in the Sedge Meadow. Actually, I did two of them, one back in 2019 and one in 2022. One before treatments and one after treatments. That study was looking at the effect of different kinds of treatments and restoration of a sedge meadow on the seed bank. And just to give you a heads up, reed canary grass in a very, again, dense population like it was in this site. I called the plant community a reed canary grass wet meadow, but there were sedge meadow plants that were still hanging on and present. That was the point of this, to restore this reed canary grass wet meadow to a sedge meadow. But I found my data showed up to 7,500 seeds of reed canary grass per square meter per about 5 centimeter depth, which was the depth of the seed bank that I sampled. So the point there is that reed canary grass can form very high and persistent seed banks. But in looking at all of these reed canary grass plants coming up in my seed bank study, I noticed, and I use this as a characteristic to help identify it, that on the ligules, especially on the younger plants, and I don't know for sure if this persists into plants this age, but it looks like to me that it might, because it looks like to me there's some pubescence right here. So I could easily see it on the young plants. There was pubescence on the, I would call it the back side of the ligule. This is the side of the ligule that's facing towards the leaf blade right here. Now, I didn't see it on any other of the other grass species that I was seeing, and there really weren't that many. There was some blue joint grass, and there was some foul manna grass, and there was some prairie cord grass in this sedge meadow, reed canary grass wet meadow. None of those species have that pubescence on the backside of the ligule. Again, I don't know for sure how much this persists into more mature plants. It looks like to me it does persist in the lower half. The upper half of it doesn't have any hairs on it on either side. But it looks like to me there could be some hairs still present right down here. When I was looking at these younger plants, the ligule wasn't this big. The ligule was only about this big. So this is representing, of course, the ligule has gotten bigger as the plants have gotten larger. Another thing I came across, and I don't know for sure about this myself because I haven't looked at this, but there was the comment that a good way to separate it from blue joint, for example, and I'll be talking about blue joint just a little bit, but other grasses perhaps, is that the rhizomes have a reddish color to them. That's what these two yellow arrows are pointing to. These are reed canary grass tillers or large stems that have been pulled up, and there's a lot of rather, again, long and persistent rhizomes, of course, in this species that makes it very vegetative in terms of its growth, of course. The rhizomes are somewhat scaly, but they also have this reddish color. And I could see it in this picture here. It's pretty faint, but there's a sort of a reddish pink coloration there. And it was the website was remarking that that's not the case for some of the other grasses that you would find in a sedge meadow. Well, I was asked to compare this with a few species, and one of them was is this species here, which is wedge grass. A couple of species of wedge grass, Spinophilus intermedia, and Spinophilus obtusata. They have a little bit of a gestalt, look. You might say that you could sort of maybe mistake these for Reed canary grass. I think they look more like June grass. Basically, the inflorescences here, the secondary inflorescences remind me a lot of June grass. If you recall again, the secondary inflorescences of Reed canary grass here, especially this form right here, which is the one that resembles the wedge grass. These are going to be thicker and more dense. Looks like on the wedge grass here, there's more branching. They're not quite as completely contracted as what we see in the Reed canary grass. This is a smaller grass too. The height of this grass is at the most about 1 meter tall. It's usually going to be more about 50 centimeters to maybe 75 centimeters or so tall. The blades of the leaves are going to be quite a bit more narrow than again on Reed canary grass. Both of these species have leaf blades, let's see, that are about 2 to 6 millimeters wide or so about 5 millimeters wide maybe on average, which again that's really at the very, very small end of the scale for Reed canary grass. But again, if you wanted to be absolutely sure and wanted to look at the flowering parts, again, the spikelets, both of these species are very distinctive in terms of their spikelets because they have a floret here with 2 glooms. Here's another floret here for this species with 2 glooms. You can see both of these spikelets have 2 flowers. There's a flower here and there's a flower here. And remember, the glooms are what subtends and sits below the spikelet. But what's unique about wedge grass is that the glooms are very, very, very different from each other. We have one gloom here that's just very, very narrow, and the other gloom is very much larger and broader. And it gets even more so with obtusata here. This one here, the upper gloom is described as being almost hood-like or hood-shaped. It's so broad. So very, very different contrasting glooms. Again, remember in Reed canary grass, the glooms are, and this is the case for most grasses, the glooms are very similar looking. Usually the upper gloom is a little bit bigger than the lower gloom. Here we see both glooms are actually about the same size. But again, they are pretty much identical in terms of their size and shape and form, of course. And that, again, is not the case with wedge grass. So now you can see that wedge grass does have a membranous ligule right here. So that would make it look similar to Reed canary grass, of course. Doesn't have that distinctive collar so much. Well, it has a collar, of course, because that collar is this area right here again, but it's not a different color. It's not set off. It's not set apart really as distinctively as it is in Reed canary grass. Another species that was suggested to take a look at in terms of similarity, and this one certainly is important, is blue joint, because blue joint and Reed canary grass live together in sedge meadows basically. And in many ways, blue joint again does look like a very small version of Reed canary grass. Here we can see the node right here, somewhat distinctive and a little bit lightly colored like it was in Reed canary grass. Here's the ligules. There is a difference. The ligules in blue joint here are usually bigger and taller than in Reed canary grass. Of course, these things all have a range that they fall within, and so there's variation. But generally, for blue joint, the ligules are, let's see, looking over here at my, yeah, so they can be up, well, they're not that much, I guess, different, up to 12 millimeters. And I think I said 10 millimeters for reed canary grass. But one thing that does seem to be more true with these is that they tend to be more torn or lacerate. You can kind of see in this one here. So the ligule probably isn't going to be a whole lot of help. What you have to look at is really the whole grass. Also, the inflorescence somewhat similar to it has at least some similarity in terms of the branching pattern. It's completely open here. It doesn't contract down nearly like reed canary grass does here. Again, so again, this is reed canary grass afterwards, after it's contracted back down. Much more tighter grouping there, a tighter compressed panicle. Whereas again, for blue joint, you don't see that at all. It remains somewhat open and spreading. You can see individual branches here. And when we look at the spikelets and the florets, very distinctive difference. So again, this is a very easy way to tell them apart if you do have the flowering parts. Here are the glooms, upper gloom and lower gloom. And so they are very similar to each other again, just as they were in reed canary grass. This shows that the glooms have some most little teeth-like or very sharp little hairs right on the median vein, basically right here, right in the center. You can see those in this little diagram here. But more importantly, what we see is that the floret has this tuft of hairs, basically. There's a very, very dense tuft of hairs that sit at the base of the floret. This is the spikelet here with just the glooms, and the floret, a single floret was in here. That floret was removed, and we can see the floret here by itself. And it has all these long hairs that come from the base of the lemma right here. This is the lemma. And from the base of the lemma, there's all these hairs, long hairs. There's just as long as the lemma, if not even longer. So very, very dense tuft of hairs. Also, the lemma has an awn coming off the back of the lemma right here. And that's something in reed canary grass doesn't have at all. So if you can get a hold of the just a spikelet here and take a look at it, even with a hand lens, you can certainly verify for sure what you're looking at. Vegetatively, you may have a clue one way or the other, but you can discern for sure if you just look at the floret. What I use a lot, and again, I've had to make a decision on whether I'm looking at reed canary grass tillers or blue joint tillers many, many, many times in doing that research in the sedge meadow, and then sorting biomass samples. Reed canary grass, again, is more robust and broader in some ways, broader stems, broader leaves, and blue joint is much more of a slender grass. Think of it as being much more slender. Also, and reed canary grass does not do this, blue joint will have this branching. There will be branches that come out of the leaf axils further up the grass here. And quite often you get towards the top of the plant, though there's a lot of leaves that are kind of all clustered together in a bit of a tuft, you might say. That is, to me, is a very good gestalt identification for blue joint. Plus the fact that blue joint does sort of have this bluish green tint to it, a little bit more so than what reed canary grass does. But I think just the slenderness, the leaves on blue joint, let's see, the blades on blue joint are 2 to 8 millimeters wide. So they are about, again, 8 is the maximum. And again, as I said, typical large robust reed canary grass is going to be 15. So reed canary grass is about twice, the leaf blades are twice as wide as the leaf blades on blue joint. Of course, you're going to have problems when you're dealing with very young tillers just coming up. That's when you can use that pubescence on the ligule. Because again, when you're dealing with just younger stems, younger tillers, things aren't completely developed, things are a lot smaller. And so there's going to be more overlap, of course, between blue joint and reed canary grass at that time. All right, that does it for reed canary grass. Another request was to, come on. Oh, one more slide, one more picture to show you again, another picture of blue joint. And again, here's everything's intact here with the glooms and a floret, single floret. And this one, the glooms have been pulled back. You can see the floret. Again, we can see, can't see quite as well, but here are those hairs. You can't see them as well here in this picture because of the lighting probably, but you can see some of the hairs here and some of the hairs right here. All right, the next one is Phragmites. So Phragmites is a native species. Phragmites australis is a scientific name for common reed, which is a species that's distributed all around the globe. In North America, we have two subspecies, though, unfortunately. We have the native subspecies, which is Americanus on the right here. And we have then the European subspecies, australis, as well, has been introduced. The non-native subspecies australis was introduced on the East Coast, probably in the late 1700s, early 1800s. So it's been in North America for a long time. And has been, of course, spreading westward ever since. It certainly is in the Midwest. You can certainly find populations. I was asked to look at a population in Ames not too long ago and confirmed it was the European subspecies. So this is another one that we need to be on the watch for, of course, because again, pretty much like reed canary grass, the European subspecies in North America is very aggressive and can form very, very dense stands. It grows probably upwards of 3 times. It can get about 3 times as big as the native subspecies. The typical height for the native subspecies is a couple meters or so, but the non-native one, this one here, can get as much as 6 meters, about 20 feet. So it can get much, much bigger. The litter that it forms from all this growth, and there's probably some ecology that explains this, of course, does not break down very rapidly. And so it just suffocates everything then that's growing there because of its own shading effect, how big it gets. But then the litter that it produces, again, as I said, is very persistent. And so it really has a negative impact then on what's growing or what was growing there in the herb layer. So there's lots of things that can be used to distinguish which of the two subspecies you have. There's actually there's some, if you go just search for it online, I'm sure you'll find a couple PDFs that are nice handouts that go through pretty much what I'm going to talk about right here. One of them was produced in Michigan. So several things to look at. Probably the most easiest thing vegetatively to look at would be the ligules shown right here. And you can see there the difference shown right here. We're talking about, again, this little ligule, which is that there's this structure that is usually a membrane, but the membrane can have little hairs or ciliate structures at the top of it. And that's the case somewhat in Phragmites. Or in other species, it can just be hairs, but it's this very unique structure, again, that you need to learn if you're going to look at grasses. In the European subspecies, the ligule is 0.1 to 0.4 millimeters. It's just a very, very narrow band. Now we're talking about the height of the membranous part without including the hairs, if there are any hairs. And usually on the European species, there are some hairs that come off the top of that. So we're looking at just the membranous part of the ligule in terms of this measurement. The native species is 0.4 to 1. So that's pretty much substantially different and distinguishable from the non-native one. 0.4 would overlap, of course, but generally you're probably going to be more than that. And this one, again, doesn't have quite the amount of little hairs that come off the top of it. Another one would be to look at the glooms if you've got spikelets. This is another thing that's very distinctively different. This is just showing the upper and lower glooms here without any florets. The florets have been removed, so we just concentrate on the glooms. And here we have measurements on the lower gloom, 2.6 to 4.2 for this one. For the native one, it's 4 to 7. The leaves on the plant, generally, there's a bit of a difference. Again, this is harder to tell, of course, unless they're kind of growing side by side. But on the European one, a little bit more of a bluish gray-green, and that's what this leaf is here. We'll compare this leaf, more of a bluish gray-green leaf, to more of a yellowish green leaf over here. And then this last thing is the lower internodes on the stems. So, most of these are vegetative. In the European species here, the lower internodes are yellowish in color. The sheath, which again, the sheath is a part of the leaf that wraps around the internode. It starts at the node where the leaf is attached and then goes up the internode. And then, of course, at the top of the sheath is where the blade comes off. Here we're looking at just the sheath here. The sheath is fairly persistent. It's pretty open here, of course, because we're looking at the internode right here. This is a part of the sheath right here, and then this is part of the sheath. Here you can see the sheath is coming together down here. But nonetheless, the sheaths are, there's some degradation here, but still somewhat persistently present there. And of the internode that we can see is this yellowish color. Whereas over here, of course, in the native species, subspecies, I should say, what happens here is the sheaths disintegrate more and are lost, and exposure of the sun then to the internodes here turns it this reddish or more of a purplish color. So that can be a pretty easy thing to look for. Another thing, of course, is just the growth. And here again, we're looking at the glooms again, just another picture. Here, I would say that both the lower gloom and the upper gloom are about half, in Australis, the European one, about half the size of the lower and upper gloom in Americanus. There is some difference usually in the inflorescence, much more densely and vigorous inflorescence in the European one versus the native one. Although I have seen the native one, secondary inflorescence looked a little bit more like this, but in these pictures we can see there's quite a bit of a difference there. Also in this picture here as well. And again, you're just going to notice that generally the native one is less dense. The stems are more scattered out. It's a native species or native subspecies in this ecosystem, and so there's a biological environmental controls that are happening there. It doesn't become overwhelming, not nearly to the extent, of course, as a really, really dense, thick patch of Phragmites. If that's what you're seeing, then it's a good chance that it's the European one. Okay, excuse me. I think that's all for Phragmites. So next one is bittersweet. This was another request. And this one is another very important one to know as well, because Oriental bittersweet is very, very bad ecologically. I'm doing a woodland restoration for the city of Des Moines, south of Grand Avenue, about 37th or so on Grand Avenue. And man, there was some gigantic Oriental bittersweet in there, along with tree of heaven and all kinds of other invasive stuff. But this is definitely a very important dual to be able to distinguish. Here we're looking at just some of the leaves pretty much, and then of course the locations of the flowers. So Solastra scandens, the native American bittersweet here on the left, its leaves are always going to be more elliptical, you see here. And from my experience, this is a very good characteristic. They always have this very acuminate tip, have this very long, slender, pointed tip. You can see it on these right here as well. Now, again, you have to look at more than just one leaf. There's variation. There's always going to be variation in these leaves. So you look at many of them and see what the general consensus of the leaves is. Whereas Oriental, or it's often called round leaf bittersweet, of course, the leaves are more orbicular, usually. But here's an example of a plant that looks a little bit more like American. So the leaves, again, can vary in shape. They can have this more elliptic, broadly elliptic look. They have a little bit of a tapering tip here, so that's going to confuse things even more. So there are going to be times when the leaves might not be very much help. So then, if you can, see if there's any evidence of flowers or fruits, and see how the flowers are positioned. Where are the flowers? Because in the Oriental, European invasive one, the flowers arise from the leaf axils. And so there's little flowers, little pedicels of course, which are the stem of the flower, pedicels arising in the leaf axils, kind of all along this branch here. So essentially what's happening there is the flowers are kind of scattered, coming out from where the leaves are attached. Whereas in the American bittersweet, the flowers are all more terminal. There's a large cluster, or again, inflorescence, probably a panicle would be the right term for it, that comes off basically the end or the top of the stems. We can see some differences, other differences in the flowers here, and there's some more pictures of the leaves. So this is it's called a polygomodioecious species. Polygomodioecious is a really big word, fancy word. Polygomodioecious means that there's some plants that have perfect flowers, meaning both sexes, and then also some other flowers that are staminate. So those are kind of male leaning plants. And then there's the other individuals that have, again, perfect flowers. So again, flowers that have both sexes, and they also have some extra flowers that are pistillate, or female. And so basically, it's sort of like you've got a dioecious species, because there's individuals that have just pistillate and just staminate flowers, but they also are, both of those individuals are producing some flowers that are perfect as well, just to add more confusion, I suppose, for us, but also more potential for seed formation. So here we're looking at the male flowers, because it's the male flowers that are helpful. In the American bittersweet, the male flowers produce yellow pollen. So if you're looking at the anthers, and the anthers are shedding pollen, you'll see all this yellow little specks and dots of pollen. Whereas in the non-native bittersweet, the pollen is white. So flowering, you can find some male flowers, pretty easy distinction there. Here's the fruits of both species. So the fruit is a capsule. The capsule over here is this yellowish orange, you might say, or it's more yellow than orange probably. And that is sort of the distinction, the color of the capsule wall. More of a yellowish color here, more of an orangish color over here. The capsule is split. That's what's happening here. Capsule is split open in order to release the seeds, and that's what's happening here. The seeds are these things here, and so they're the same. There's really no difference between the two species with in terms of the seeds. What the seeds are covered by a structure called an aril, A-R-I-L. An aril is a fleshy outgrowth from the seed coat that, of course, makes the seed more enticing for seed eaters and fruit eaters to consume, of course, and that's the way it gets dispersed, of course. So there is a seed here, but it's completely encased in this fleshy red tissue, which is the aril. And these are pretty persistent. They last through the winter time. So again, it's a very good food from that standpoint ecologically for birds, because it does provide food source when it's harder to find food. But it also means, of course, that both species can get spread pretty easily by birds. The other thing that I'll mention that I've seen from my experience is that again, the Oriental bittersweet is it's capable of getting much bigger liana. These are lianas, which of course means it's a woody plant, but it's a climbing woody plant. So it climbs up on other species, primarily trees, of course, climbs on trees, and that way it doesn't have to produce as big of a stem to support itself, but yet it can climb up into the upper canopy and get access to light. So they're both lianas, but the Oriental one can get much bigger. It can get like 40 feet long, a liana 40 feet or more long, and the stems can be pretty darn big. They can get to like the size of a small tree. And this is showing the bark of the Oriental, of course, here. Whereas the bark and the size of the native bittersweet is the size is much smaller, usually up to 15 feet or so, maybe it would be the total length of a plant. And of course, the bark has a much smoother look to it, and thinner bark. All right, the next one was also requested, and this is a good one too, because we do have a lot of problems with Siberian elm, Ulmus pumila, distinguishing it from the native elms. So this slide shows everything you see here is a Siberian elm. And so these are obviously leaves here. Well, a few pictures of some leaves. These are the samaras, the fruits. Samara is a type of a single-seeded winged fruit. So there's a single seed right in the center here, and then this wing that's an outgrowth from the pericarp wall that surrounds the seed. And we can see some buds over here, and we can see some bark. So several characteristics here. And then the next slide here shows the two species of native elm, American elm and red elm. And the thing that the Keys mostly use to separate Ulmus pumila from the native elms is the size of the leaves. And of course, I don't have anything for scale here, but you can kind of get a sense that these leaves are smaller. Basically, if the leaf blades, I'm just looking at the blade itself, not the petiole, just the blade, there's not too much of a petiole on these. There is a little one, but you'd be measuring just the blade, and the blades on Siberian elm are generally always going to be less than 7 centimeters or about 2.5 inches. So the leaves are distinctively smaller, basically, than the native elms. On the native elms, they're longer than 7 centimeters. 2.5 inches, and can even be more than that, of course. Of course, now the problem of looking at a very young seedling sprout, a plant that's just a couple feet tall, that's going to be a problem, because now you're going to have smaller leaves. Whatever kind of elm it's going to be, it's going to be smaller. The leaves are going to be smaller because the plant's smaller. And there's always going to be, the possibility of an individual that's just not very big yet. It could be pretty difficult, especially using the size of the leaves. But there's some other things. Now, on the native elms, one thing that you'll read about them if you look at anything that talks about leaf morphology, they're usually described as doubly serrate. And so doubly serrate means that we have these big teeth that we can easily see along the edge, and then there's tinier teeth that are on top of the big teeth. So there's like two scales of teeth. Doubly serrate means that there's some big teeth, and then there's some smaller teeth that are run along the margin as well. And again, as I said, sit on top of the larger teeth. For the most part, in Siberian elm, we don't see that double serration. It's usually just singly serrate. So again, variation, but more of the leaves will just have a single large tooth. There won't be that smaller double tooth. The other thing that elms are really known for is, of course, the oblique leaf base, easily seen in these two pictures of American elm over here on the left and red elm on the right. You can see how the leaf base is not symmetrical, it's asymmetrical, and the term oblique is used to describe that. The two leaf edges do not meet together here. They're offset, basically, as you can see here. So again, that's oblique leaf bases, very characteristic of elms and also hackberries. And again, that's a feature that you don't see very well developed. Now here's one that shows a little bit of an oblique base, but most of these other leaves, as you look through these, they're not very oblique at all. And so weakly oblique at the best. There is a difference in the size of the buds too. That's why I put this picture of the buds in here, and also there's pictures of buds over here as well for both of these species. And it's again a difference on size. So no, these are going to be the buds as measured before, like right now, before they're expanding and starting to break open. And basically the separation here is 3 millimeters, basically. So the buds over here, less than 3 millimeters in length for Siberian elm, and for the native species, they're greater than 3. A really good thing, if you have the fruits, is the samaras. On the Siberian elm, the samaras are just completely glabrous. There's no hairs anywhere on these. I think there's a chance you might find some like very, very minuscule hairs on the edge of the samara, I believe, but pretty much there's just simply no hairs anywhere here. On both of the native species, and this is how you can distinguish the American elm from the red elm very easily when it's dispersing these samaras, is you can see that the American elm is very conspicuously ciliate, meaning little short cilia-like hairs coming off the margin of the samara here. Very conspicuous, very easy to see. Those are missing. Those are not present on red elm, but red elm is hairy over the area of the samara where the seed is. Right in this area, there's some pubescence, which again is not present on the samaras for Siberian elm. And by the way, if you're looking at just the twigs of these two species, the buds again are over 3 millimeters long. That separates them from Siberian elm. But there's a difference. The red elm here has some copper colored hairs that come off of the bud scales especially the upper bud scales. We can kind of see them right in here and right in here. Those copper colored hairs. And then over here, the bud scales are pretty much glabrous. There's no copper colored hairs coming off any of these. Now, the thing that I use almost all the time to distinguish red elm from American elm out in the field, because these samaras come in the early spring before leaf out. And so they're gone quickly, soon after the, again, seed dispersal occurs. So you can't use these samaras except for a short period of time early in the spring. You have to use the leaves. And there really is not very good ways of using the leaves, other than there's a tendency for red elm to have these forked veins. Some of these veins are forked right here. So you can see the arrow pointing to these forked veins, whereas you generally don't see the veins forking in American elm. So you could look at the leaves, and that would be a helpful hint. Both of them are very rough and scabrous to the touch, so I don't think that's a very good characteristic to try to use. And so what I'll often do, of course, is I'll take out my pocket knife and take a look at the bark. And basically, here's the bark for Siberian elm. Very, very strongly ridged and furrowed bark. You see here, this picture here. And what you do is you just take your knife and you slice down the for the edge of one of these ridges to take a look at the layers and colors of the bark below the surface of this ridge. And in Siberian elm, which is what this is again, it's a very, it looks a lot like walnut, basically. It looks a lot like black walnut, a very sort of deep brownish red color all throughout this area here, all throughout the bark tissue here, cork tissue. Whereas over here, American elm will have white and red layers. So this is looking at it a little bit differently than I was showing you in the other picture, but you can still see this if you do that slice, like I described on the previous slide there for Siberian elm. But you can usually see some reddish and white layers, very, very distinctive reddish and white layers. And the white layers are pretty large. And in fact, more white than there is red, basically. You can sometimes see this difference on the external surface of the bark too, but I don't have a picture of the bark here. But whereas in the red elm, it's all reddish pretty much. There isn't any really white layer in through here. Now, again, this is going to look then more like Siberian elm, but I think there is even a difference here because, again, this doesn't have quite that deep, rich, reddish brown color that we were seeing with Siberian elm. What's next? Let's see. Okay, I threw this one in. No one asked about honeysuckle, but I thought I should quick throw this in because honeysuckle is so terrible. And you need to, again, be aware that there's some things that look like honeysuckle, of course. And what I have here are the two most common invasive non-native honeysuckles, Lonicera mackii, Amur honeysuckle up here, and then Tatarian honeysuckle down here. And again, so what makes it look like these species over here, of course, are the simple opposite leaves. Dogwoods have simple opposite leaves. That's what we have up here. And then there's a couple of native species of honeysuckle down here. Now, the native species are really easy to separate from the non-native species because the two native species of Lonicera we have in Iowa are both lianas. They're both vines. So they're, no, excuse me, I misspoke. We shouldn't call them vines because that's the term we use for herbaceous plants. They're climbing woody plants. But and so they also have the opposite leaves, of course, that Loniceras have. But the other very distinguishing feature on the native Loniceras here is that, you'll see these opposite leaves throughout much of the plant. But when you get towards the top of the plant, you'll see the last pair or last couple pairs of leaves are basically perfoliate. They are joined at the base, like in cup plant, basically. and the stem kind of comes through then those fused leaves. That's what perfoliate means. And we see that in both of these, Polyphora and Dioica. They're both going to have those fused leaves towards the top of the plant. So very easy to separate, of course, from the non-native ones. And then the non-native ones have hollow pith. So if you're able to take a look at the stem and just slice it longitudinally or cut through it, you can see that hollow pith right here. The flowers on the non-native honeysuckles occur in the axils of the leaves. Basically, there's a pair of flowers. Now, these are two flower buds, haven't quite opened up yet in a mure honeysuckle, but there's a pair in this leaf axil, there's a pair in this leaf axil. And here we're looking at Titarian that's gone to fruit. And so the fruit, of course, is also going to be pairs of berries in the leaf axils. One difference between these two, other than the leaf shape, is that the fruits in a mure honeysuckle, and I should say the flowers in a mure honeysuckle, are sessile, whereas the flowers in a Titarian honeysuckle have little pedicels that are longer than the petiole of the leaf that they are in the axil of. Here, again, you can see there's really not any pedicel at all. Well, actually, it would be a peduncle because there are two flowers here, and so very short or non-existent peduncle right here, certainly shorter than the petiole of this leaf right here. Down here again, that peduncle would be longer than that petiole. So that's very different from the dogwoods, of course, over here that have these clusters of flowers. The clusters of flowers come off the branch tips, come off of the tips of the branches in a very large cluster of flowers. And there'll also be a cluster of fruits, and they're called drupes in the dogwoods. But of course, you can always look at the leaf. And of course, these two leaves look pretty similar. Same shape, opposite, have an acuminate tip to some extent here. The veination in the dogwoods is going to be maybe a little bit more arcuate in terms, again, we talked about this in the shrub webinar. The veins come out and kind of curve and follow the margin of the leaf somewhat. And you can usually see that best looking at the bottom of the leaf. But of course, you can always do the test by just tearing the leaf somewhat with your hands, just tear the tissue apart and look for these threads of elastic tissue. Basically, it's material from the phloem that has solidified and was exposed to the air, but these sort of elastic little threads that join the veins of either side of this torn leaf. You can see one there, and one there, and one there, and one there. That, of course, is the conclusive evidence of dogwood. You're not going to see that with the honeysuckles. The next one was I had sent this is a slide that you don't have. I had gotten the presentation ready and sent it out to Lance. And then I looked at some emails, and it was an individual who had emailed me and said that he had some trouble identifying buckthorn, European buckthorn from wild plum. And so I quick threw this slide in to show European buckthorn here, which again is a very, very nasty invasive species from a couple of potential lookalikes, I suppose you might say. So American plum up here in the top. And of course, then there are native buckthorns. This is the native buckthorn that you would be most likely to run into. There's some other ones that are native, but they're very, very limited in their distribution in Iowa. This one's Rhamnus lanceolatus. It's pretty much throughout Iowa, but it's not real common. So it's nothing that you don't see it a lot, but you certainly could find it in most places. And so those are the two lookalikes to compare with. And so buckthorn over here, here's a twig with the leaves, and you can see very easily, of course, that the leaves are opposite. They are described as being opposite or sub-opposite, and sub-opposite just simply means that they're not perfectly opposite, then they're almost opposite each other. They're joined not quite at the same location on the twig, but very close. And here's a couple of separate pairs of leaves shown separately, of course. And so the shape of these buckthorn leaves is somewhat somewhat orbicular. They can be roughly about the same length, and width can be about the same, maybe just a little bit longer. Generally, the length is about 1 to 2 times more than the width. And this one here is probably about 1.5 times more. So this is a good, shows the range potentially pretty good here. Some of these up here are probably more like these on the bottom right here. So that's pretty important. Also, the really glossy, kind of shiny, dark green coloration that we see in the buckthorn leaves is important and is helpful. The veination is somewhat like in the dogwoods, again, with these veins that are kind of arcuate, which again means they come out and then they kind of go parallel to the margin of the leaf, somewhat, rather than running right to the margin of the leaf. They don't do that. They come out and they follow the margin of the leaf for a ways. And then it's called buckthorn because of the presence of some thorns, but the thorns are basically present at the tips of the twigs. So that's where you're going to see the thorns in buckthorn, coming off the tips of twigs. And again, a thorn is a modified branch. So that's the correct term for this right here. Now, I could see why you might, again, sort of look at American plum and be kind of confused, because American plum also has thorns. But these thorns come off all along the twig. These are several thorns coming off all along the length of the twig right here, and not just at the very tip. Of course, the other thing about American plum, of course, is that the leaves are alternate, not opposite, and they're much longer and more elliptic. Now these are elliptic right here, so that's elliptic. This is elliptic as well, so there's some overlap there, but again, the leaf arrangement is totally different. Alternate leaves here, opposite leaves here. And remember that in many of the prunus, there are glands that sit right at the top of the petiole. This is the petiole, right at the top of the petiole, right where the petiole and the blade meet. A lot of prunus species have these glands. Prunus americana, the American plum, probably has these glands less often than it does have them. So the description for it is that it's usually eglandular, eglandular meaning lacking glands, but they still can be present. But this would be important, again, if you see these glands like this for any prunus. And of course, then in Rhamnus lanceolate, again, the leaves are alternate, which again is very distinctly different from the European buckthorn with the opposite leaves. And these leaves are much more lanceolate or elliptic shape, longer, much longer than broad compared to the European non-native one. So that should do you for buckthorn. And then I had a request to look at Lythrum salicaria, purple loosestrife, especially with respect to identifying it from vegetative lookalikes. It's pretty easy when it's in flower. If you take a look at a flower book or anything that shows a picture of the flowers, you can see some flowers right here in this plant. So I'm not going to try to use flowers here. The idea is to do it vegetatively, because that's when this individual is scouting for this species in the individual conditions. So, excuse me, when they're not in flower. So we see Lythrum then here on the left, and so it's got opposite leaves. That's an important characteristic. It's got leaves that are pretty much sessile. Here's some leaves off separately by themselves. There's no petiole down here at all. There can be a tiny, tiny short little petiole, maybe in some situations, but most of the time not. Pretty lancelet shaped, as we can see as well. The stems are somewhat squarish, so somewhat four-sided to maybe six-sided or so. There can be just a tiniest hint of maybe a ridge, but they're really not. The ridges on the stem are not well developed at all. The fact that it's got 4 to 6 sides sort of makes this corner then, but there's really not any kind of a well-formed ridge associated with that corner. So one thing that this looks a little bit like then with the opposite leaves, oh, also the leaves are decussate usually. Decussate means that a pair of leaves at this node are going to be perpendicular to the next pair of leaves, either up or down. So then this next pair of leaves up this way are turned at a 90-degree angle to these leaves right here. And that's going to happen basically when you have sort of a square stem. So those all are characteristics that are very mint-like in the mint family. And they also are very similar in some regards to swamp milkweed. So swamp milkweed is certainly a species that could be mistaken if you're not paying attention. You can see you've got these opposite leaves, of course. They're also somewhat decussate here on this individual, arranged again at 90-degree angles. And somewhat longer, maybe a little bit more narrowly lanceolate, but nonetheless have a similar veination pattern. See these veins here and these veins here. So what's the difference? Well, obviously, hopefully you would think of the one thing, of course, with the milkweed is that you can tear this leaf a little bit here and you should be able to see some milky latex. Milky latex is certainly present in swamp milkweed. The only milkweed it's not really present in is butterfly milkweed. And the other thing is that swamp milkweed is going to have a petiole, pretty well developed petiole. You can see it right here on this leaf right here. Usually the petiole is at least a centimeter, probably can be as much as 1.5 centimeters in length. And that's a very clearly petiolate leaf then. And we're not going to have that on purple loosestrife. Another one that, again, just glancing at would look somewhat similar as obedient plant, Physostegia. It is in the mint family, of course. So now, again, we do have those overlapping mint characteristics, that square stem, the opposite leaves. But we can see very easily here, again, on this one also, too, is sessile, just like the purple loosestrife is. There's no petioles really here at all. But the difference, of course, we can see is that this is serrate. Purple loosestrife has got leaf margins that are entire. There's no teeth, and that's true for the entire family of Lythraceae, is entire margins on the leaves. So the serrations that we see here, and of course, they're not strongly developed by any sense of the imagination, but certainly easily seen there, are going to make this identifiable as not being purple loosestrife. Here we can see the teeth pretty well developed down here on this leaf. Well, there's some other species as well that other mints and other species that are somewhat of a lookalike. Another one here is monkey flower on the left, Mimulus ringens, which used to be in the Scrophulariaceae, but it's now in the Firmaceae. So you can see again that too, it's got the sessile leaves, it's got opposite leaves, sessile leaves. There's the teeth are not really well developed on this plant. So again, you could look at this and think that it's somewhat entire, or it looks like it's kind of entire, because the teeth, not really teeth, it's more of what's called scalloped. Just a little bit of a scalloped margin there. But what is more helpful here is that these leaves are clasping at the base. Down here at the base, these leaves are clasping. They have little auricles that somewhat clasp around the stem. You can see the auricles right here. The stem would go right here in that center part right there. Those auricles kind of clasp around it. And remember we said that the stem on purple loosestrife is not really ridged at all. It's bluntly squarish or bluntly hexagonal. But usually there are some conspicuous ridges that descend from the base of these leaves. You can kind of see a ridge right there. Again, obviously these things in flower, no problem separating them from purple loosestrife. We're focusing on what these the situation would be vegetatively. The one in the center is marsh mint. Mentha arvensis is the old name. Mentha canadensis will be the new name when Flora North America comes out in the mints. This one, so again, has just a general sort of gestalt because of the opposite leaves and the square stem. But and if you didn't have the flowers here, then it would look a little bit more like it, I suppose. But Mentha arvensis, of course, has vertisols, flowers in the leaf axils, and it's got very, very conspicuously teethed margins on the opposite leaves. The leaves are going to be more short, not nearly as long as the leaves on purple loosestrife. And this thing smells like a mint, just if you're even walking even near it. You can just slightly crush any of the tissue, you will smell the mint from marsh mint, which of course is not anything you're going to smell with purple loosestrife. And another one in the mint family is Stachys. This is Stachys pilosus, which is basically the correct name now for what you might still be calling Stachys palustris, which is not native to North America. And as far as you know, Stachys palustris is not in Iowa. But Stachys pilosus is the most common, probably hedge nettle, marsh hedge nettle that you'll find in wet areas. All of these things grow in wet areas, the same kind of place, of course, where purple loosestrife grows. But again, we have very distinct differences here. Very, very hairy stem. The leaves are very toothed, they're scalloped. They're certainly not entire margins. Again, just gestalt-wise, there's lots of things that look quite a bit different. The leaves are, again, aren't as long and narrow as in purple loosestrife. But good to have some pictures. All right, then the last thing that was requested, about 15 minutes, I guess, was just some of the spring ephemerals and spring blooming species. And I thought, well, this would be a good way to end anyway, because hey, it's spring is right around the corner. We're already starting to see some of these species bloom, of course, and we'll be seeing more when it gets back to 60 degrees or so later this week. So why not do just a quick review of some of these spring blooming species and spring ephemeral species. Now, of course, there's a difference between spring ephemeral and spring bloomer. A spring ephemeral is a spring bloomer, but a spring bloomer is not a spring ephemeral. This one was requested because they're both Cardamine species, spring cress, which is a species you'll find in wet areas and along marsh edges or sedge meadows. And then Cardamine concatenata, which is the toothwort, which you'd find, of course, up in upland forests. This one is a spring ephemeral, but both of these are spring bloomers because they bloom in the early spring. Spring ephemeral, of course, is a plant that does bloom in the spring, early in the spring, but it does all of its annual growth, its entire perennial life cycle, basically, in just a few weeks, probably about 4 weeks or so. Comes up, gets some photosynthesis going, starts forming the flowers, starts getting pollination on the way, get seed formation underway, get fruit formation underway, get these fruits down here and seed dispersal. all within about 4 weeks before the canopy closes in overhead in a forest or woodland environment. And then the plant completely senesces and dies away. And there's no evidence of it pretty much after June 1st or June 15th or so. It is back to hibernation, or I guess we should say estivation in the summertime. It's gone dormant and it is just resting below ground in its perennial tissue, usually a tuber or a rhizome or a bulblet of some kind. And it'll be there, of course, for 11 months or so until the next spring. So that's what a spring ephemeral is, and that's what a few of these are. So the difference between toothwort and spring cress is pretty obvious in the leaves, right? The flowers are very similar. They're both mustards. They both have 4 white petals, very typical white flowered mustard. So those aren't going to be much help, but just looking at the leaves here are very much different than the compound leaves of toothwort, these divided leaves here. Trifoliate leaves. So we've got a leaf that's divided into 3 leaflets. And the leaflets themselves are very, very deeply lobed. They're not quite cut down to forming another leaflet, but the leaflets themselves are deeply lobed. The next one is the hepaticas. And again, depending upon who you use for your taxonomy, Flora of North America now has separated Hepatica nobilis into 2 species and put them in with the anemones. So we've got anemone acuta the sharp-lobed Hepatica, and then anemone americana, the round-lobed. And so the Hepaticas are these really distinctive plants because of the basal leaves having these 3 very distinctive lobes. The leaves are not toothed or anything. The margins are entire, but there's very deep lobing happening here to form very distinct 3 lobes. And so the difference, of course, in Acuta versus the Americana is the tips of those lobes. We have lobes over here that are fairly pointed, clearly come to a pretty well-defined point, more of an acute type of tip, and lobes over here are very much rounded. So 2 different species. Unfortunately, BONAP does not recognize the 2 different species, and they call them both the same species still under Hepatica. So the distribution of these is a little bit hard to figure out, but for the most part, this species is by far the more common one. Sharp-lobed occurs pretty much from if you drew a line from Kossuth County down to Ringgold County and to the east in Iowa, it's pretty easy to find in woodlands and forests that direction. Now, these are spring bloomers. They're not spring ephemerals because you can find these leaves all through the growing season, still present and still doing photosynthesis. So they're just taking advantage of the sunlight early in the spring with the lack of a canopy to do flowering. They're in the Ranunculaceae, the buttercup family. So what we're dealing with here are sepals rather than petals. The petals are absent. They have 3 bracts. They have some bracts here underneath petals, but lots of stamens, lots of separate simple pistils or carpels. Flower color can vary somewhat from white to bluish. And by the way, these leaves here are the leaves you see when it's in flower in the spring. The leaves will persist during the wintertime over winter, but they turn more this maroon purplish color and start disintegrating basically. And sometimes when the plants are blooming then in the spring, they're just beginning to disintegrate or starting to die back, or they maybe have already completely died back and might be gone. So you may see plants that are blooming without any leaves, or you might see some that are blooming with leaves that look kind of like this. Then, of course, the new leaves come out after blooming and look like these green ones here. Then we see a couple trilliums, the snow trillium, which is blooming right now. And this is a spring ephemeral, pretty distinctive little plant with the three petals, then the three sepals right below it, then the three leaves. So and it's just got these three whorled leaves that are down below. These three leaves right here, you see right below the flower. So the three leaves and the three sepals and the three petals. Small little plant, about maybe 6 inches tall or so. Again, with the first things to bloom in the forest. And this is another trillium that will be coming along soon. It's not quite as early as snow trillium. This is Trillium recurvatum, prairie trillium, or wake robin. Snow trilliums occur pretty much throughout the entire state. They go all the way over into western Iowa. They do not, well, they barely make it across the river into eastern Nebraska, just right along the Missouri River. They do, I don't know for sure that there's very many of them in the counties, western Iowa counties along the Missouri, but I know they are, like in Crawford County, for example, which is the next line of counties to the east. So they're pretty close to being statewide. Whereas recurvatum is very definitely an eastern Iowa species. It is pretty much in the eastern, I would say 1/3 or so of Iowa. Not so much all the way up in northeast Iowa, but starting from about Dubuque County going south. And there are populations in central Iowa. I have found this species in some of the Des Moines parks. So it comes as far west as Polk County. That would be pretty much pushing it in terms of its western extent, as about as far west as it goes. And it's got, again, three leaves like the other trilliums, and then a much different looking flower, of course. This has a diagram showing you the sepals here are pointed down, the green sepals, and then the three petals. Very pretty plant. All right, then we have the erythroniums. And we've got three erythronium species here. The very common white trout lily, the much less common. It used to be combined with the white trout lily, but now it's a separate species. Prairie trout lily, which also has white flowers. And then this is one is either endangered or threatened. I think it's endangered in Iowa. The yellow trout lily, which only is up in far northeast Iowa for the most part. So white trout lily, of course, occurs pretty much everywhere in Iowa. It goes across the entire state into eastern Nebraska. The prairie trout lily, if you look at the BONAP map for it, it's probably not quite correct. There's quite a few counties indicated, but as far as the Iowa Natural Areas Inventory database shows, it's really only in about the southern half of Iowa. And it looks, of course, similar, but some of the main features to use to separate are the lack of very distinct brown blotching in the leaves. That's a pretty good characteristic. The tepals, which are, again, a term for the petals and the sepals, which look alike, in the white trout lily, under good flowering conditions, are going to be recurved like this, like you see here, but not so much in the prairie trout lily. They don't recurve nearly to that extent. They kind of just go more horizontal. And then another thing that's pretty clear is when it goes into fruit and it's forming these capsules. So here's the long scape, the long peduncle for the flowers, of course, which rise from the base of leaves. This plant does not have any upright stem. It has just these basal leaves and then a peduncle that comes up from the base of the plant with the flower, with a single flower on it. And so the fruits that form here from the flower will bend over, the peduncle will bend over, and the fruits will actually lie on the ground on the prairie trout lily. That's not the case in the white trout lily. Yeah, those are the main features that help to distinguish those two species. Of course, the yellow one is obviously got yellow tepals and it has the blotched leaves just like the white trout lily does. Then the next one, next spring ones are going to be ruin enemy and false ruin enemy, two species that look a little bit alike. Both of them in the buttercup family, but different genera, of course. So what's happening in ruin enemy is kind of complicated. We look at this and we think these look like there's 6 whorled leaves here. But technically speaking, those are not leaves. Those are involucral bracts that look very leaf-like because they subtend the flowers here. And there's actually 2 involucral bracts rather than 6. There's 2 involucral bracts. Each of them are compound, divided into 3 parts. Hence the 6 involucral bracts here. There is a basal leaf, the true leaves. There are no calling leaves. So these aren't really leaves. And there's lots of websites that make that mistake by calling them leaves. The true leaves are only basal. The basal leaves are down here. And the basal leaves are trifoliate, compound, and split into 3 leaflets. And then the flowers, in terms of separating from false ruin enemy, the flowers are pretty much, seem to be always pretty consistent, always like this. There's usually always three flowers, 2 that are shorter, and on the lateral sides of a central, taller, and somewhat larger flower, as you can see in this picture. Whereas in false ruin enemy, the flowers, and this is coming off the very top of the plant, coming at the top of this scape, basically here. Over here in false ruin enemy, the flowers come out of the leaf axils, and they can be singly, there can be 2 or 3 of them in a single peduncle, so that varies. And over here, there's both basal leaves and calling leaves, and they both look something like this right here. They're both twice ternately compound, which means, again, the leaf is divided first into this segment, and this segment. Then each of these segments is divided into 3 leaflets themselves. So that's twice ternately compound. Both the basal leaves and the leaves coming off the stem alternately, the leaves on this are alternate, look pretty much like this, twice ternately compound. And one thing that's sort of different from these leaf lookalikes over here in ruin enemy is that these leaflets are much more deeply divided. You see the lobing, the incision making these lobes here goes much deeper down into the leaf tissue on these than any of these over here. There's just very, very slight lobing around the margin of these bracts, involucral bracts, which look like leaves. And then I think this is the last, no, we've got 2 more slides. Just about done. We got, and these are pretty common ones. So everyone knows Dutchman's breeches, I think, probably, right? And this is this lookalike, Dicentra canadensis, squirrel corn. Basically, the leaves are identical. You're not going to separate these at all by their leaves. They both arise from little bulblets in the ground. You see some right here for Dutchman's and these over here for squirrel corn. But of course, it's the difference in the flowers, the more pointed corollas right here and the more rounded corolla lobes right here, which makes the difference between this looking like a Dutchman's pair of pants and these looking like corn. Both of them have eliosomes. This is the capsule, the fruit that develops from the flowers, and the capsule splits open, and we've got these seeds with these eliosomes, which are again a fleshy tissue that or appendage that comes out of the seed coat that's attractive to ants. And many of the spring blooming woodland species have eliosomes, of course. The same is true for the eliosomes here on bloodroot. And there's also eliosomes on the seeds of spring beauty. So the last 2, oh, and just by the way, so again, the spring beauty is a spring ephemeral. I didn't go over that. These 2 are spring ephemerals. These are both just spring bloomers. They're not spring ephemerals, they're spring bloomers. These are all spring ephemerals. All 3 of these are spring ephemerals. We did, and I did this one. This one's a spring ephemeral. This is a spring bloomer. Though White Robin will, it will persist for a while, but it will eventually degrade. So it's kind of like Virginia bluebells. It's not quite as much of a spring ephemeral because it does do some photosynthesis for a while after reproduction is done, but it won't last the entire growing season. And then Bloodroot is a spring bloomer. And I guess I'll quit with that. 8:32.
We did a couple of questions in the chat. Someone was asking about hybridization of uh bittersweets.
Bittersweets.
Yeah.
I haven't read or seen anything that they hybridize. So I guess what I'd say is, as far as I know, the answer to that is no. And unless someone else has actually seen something about that, I've what I was looking at, I don't think Flora of North America is done for it. So that's always a good source to look at in terms of hybridization. I don't think, let's see, it might be. Yeah, Celastris is done, I think. Well, let's see. It doesn't say anything about hybrids on Flora of North America website page. Uh, I mean, as far as I know, they don't, but I haven't looked at that real close, which is probably good.
And yes. MJ was wondering how to tell the two native Lonicera apart.
Oh, the two native Lonicera apart. By the leaf shape, um, I could go back, I guess, real quick. Oops. Yeah. Unfortunately, you don't see it very well here because I wasn't trying to show enough to show how to separate these two. But the leaf shape will be pretty good to separate prolifera from dioica. And by the way, prolifera now is going by reticulata. Well, again, this hasn't been confirmed by Flora of North America, but that seems to be what will probably happen. So the leaf shape in dioica here is much more lanceolate. You see these lower leaves again, looking at the lower leaves, not the two at the very end, because those are perfoliate and joined, and so you want to look at the leaves that are lower down on the stem. Whereas these leaves, I don't have a picture of it, but these leaves are more orbicular, more round in their shape. There might be something with the flowers too, but looks like these are more yellowish and these might be more whitish, but I'm not 100% sure about that. I know the leaf shape is good.
That's all we got in the chat.
All right. Well, again, thanks everybody for the 4 webinars, and hope there's been some value for you. And it's time to get outside now, I guess, and look at things in real life rather than on a computer screen.
Yep.
Thanks, Lance.
Yep. Thank you, Tom.